My name is Haley Coonley, and my study is evaluating environmental gradient along eastern Lake Michigan. I'd like to thank the help of Megan Mader, who is a graduate student studying a similar topic, and Carla Reitz for being my mentor. I would also like to thank Grand Valley State University and Annis Water Resources Institute, for this study would not be capable. Freshwater river mouths provide unique habitats in large lake systems and are a focal point of human activity. A large lake system is composed of three primary habitats: large lake, river mouth, and upstream tributary. As you can see in the example above, Lake Michigan is a large lake, Muskegon Lake is a type of river mouth that is connected to Lake Michigan, and the Muskegon River is the upstream tributary that flows into Muskegon Lake. A drowned river mouth is a protected lake-like habitat that connects a tributary to a large lake. Drowned river mouths are resource-rich areas that provide important fish habitat, including for migratory species such as Chinook salmon, yellow perch, and lake sturgeon. In fact, fish diversity is greater in drowned river mouths than nearby areas in Lake Michigan. The importance of resource-rich drowned river mouths to Lake Michigan food web is likely to increase over time because Lake Michigan is undergoing a process of nutrient depletion due in part to invasive mussels. River mouths provide a rich array of ecosystem services that also benefit people, such as nutrient retention, recreation, resource richness, and habitat. River mouths are more resource-rich than their upstream tributary or connected large lake, which makes river mouths important habitats for biota and large lake systems. Despite their uniqueness and importance, our ecological knowledge of river mouths is much less than that of the upstream tributaries or nearby waters of large lakes. Sampling locations are selected among 29 drowned river mouths, ranging from New Buffalo in the south to Charlevoix in the north, along the eastern shoreline of Lake Michigan. The white boundary, as you can see in the photo here, indicates the watershed, and the light blue, you can see on the edge, indicates the drowned river mouths. The red triangles, the outer edge, are drowned river mouths we sampled. The 12 drowned river mouths we sampled were divided into 3 tension zones: north, transition, and south, based on latitude and eco-provinces. The tension zone is defined by the change in vegetation from eastern broadleaf forest to Laurentian Great Lakes forest, divided approximately by Highway 46 and extending 30 miles north and south. The drowned river mouths we sampled range from North Betsy Lake to Pentwater Lake. It's right there. In the transition, Stony Lake to Lake Macatawa, and in the south, Kalamazoo River. Our overall goal is to better understand the ecology of drowned river mouth habitats along the eastern shore of Lake Michigan. Our objective is to determine whether environmental gradient exists among drowned river mouths. Based on previous research, we hypothesized there is a latitudinal gradient in productivity, meaning nutrient-rich waters are highly productive, which has important implications for the Lake Michigan food web. To test our hypothesis, we measured chlorophyll-a concentration, which is a measure of phytoplankton biomass which forms the base of the food web, and Secchi depth, which is a measure of water clarity, where larger values are associated with larger water clarity. We hypothesized that chlorophyll-a concentrations will be higher and Secchi depth lower among southern drowned river mouths, meaning more productive, compared with northern drowned river mouths, meaning less productive. To sample chlorophyll-a, we followed standard protocols. Surface sampling involved collecting 1-liter water samples at 3 sites on each lake, which were at the deepest hole, the channel, and the river mouth. Bottom sampling involved using a Van Doren water sampler, 1 meter above the lake substrate. 1 liter of water was collected for each bottom sample at the same 3 sites surface samples were collected, resulting in 6 water samples per lake. The 1-liter chlorophyll-a samples were stored in amber bottles in a cooler on ice to prevent light penetration that may have skewed our results. These photos illustrate the mechanics of the Van Doren. You can see that the ends separate from the body and clasp together. The string lowers the Van Doren in the water, and the small orange and black marks count the meters in depth. Once 1 meter above the substrate, a metal rod is sent down on the string that collapses the ends of the Van Doren, holding the water sample. In the photo to the right, I am transferring the water sample into my 1-liter amber bottle, where it is then placed in a cooler. In the Steinman lab at Annis Water Resources Institute, the samples were filtered directly after sampling events. These filters were then placed in a centrifuge tube covered in aluminum foil to to decrease light penetration and stored in a freezer. Once all lakes were sampled and filtered, the samples were then grinded and extracted following standard protocols. To grind our samples, we used the machine you see to the left with a buffered acetone solution. Samples were then refrigerated for 24 hours for all chlorophyll-a to fully extract. These samples were then ran through a centrifuge. A blank sample of the buffered acetone solution was also ran through to provide quality control. Samples were then analyzed with a spectrophotometer, as you can see in the photo to the right. Each sample was analyzed twice. The second analysis involved acid being added to the sample. The photo on the left shows the Secchi disc we used for sampling. The photo on the right illustrates how to use the Secchi disc. Once the white on the disc disappears in the water, as I pull the disc back to the surface, the meter marks on the string are counted to acquire the depth of clarity. In this scatter plot, we have our mean chlorophyll-a on the y-axis and mean latitude on the x-axis. The mean chlorophyll-a is calculated from the 3 surface samples taken from each lake. The error bars are the standard deviation for each lake. You can see in the trends of the graph that lower latitudes have higher chlorophyll-a and higher latitudes have lower chlorophyll-a. The R value, shown up here, shows a negative correlation, meaning chlorophyll-a goes from high in the south to low in the north, as latitude goes high in the north and low in the south. The p-value is less than 0.01, supporting our hypothesis. This is the same scatter plot as we just saw. Again, mean chlorophyll-a is on the y-axis, and mean latitude is on the x. This plot, although it excludes Lake Makatawa, Makatawa is not erroneous. We just wanted to show that with it being the highest value, it does not drive the plot's relationship. In fact, the plot shows a stronger negative relationship without Makatawa, with the R value closer to -1 and an even smaller p-value. In this scatter plot, I have the mean Secchi depth on the y-axis and mean latitude on the x. Mean Secchi depth was calculated from the 3 sites measured at each lake. The error bars are the standard deviation for each lake. You can see in the trends of the graph that lower latitudes have lower water clarity and higher latitudes have higher water clarity. The R value shows a positive correlation, meaning high water clarity coordinates with high latitude and low water clarity coordinates with low latitudes. The p-value also supports our hypothesis. This is the same scatter plot. Again, mean Secchi depth is on the y-axis and mean latitude is on the x. This plot, although excludes Portage Lake is not erroneous. We just wanted to show that with it being the highest value in the north, it does not drive the plot's relationship. In fact, the plot shows a stronger positive relationship without Portage Lake, with the R value even closer to 1 and a smaller p-value. In conclusion, we hypothesized there is a latitudinal gradient in productivity among drowned river mouth lakes along eastern Lake Michigan, and this is supported by our mean chlorophyll-a data and our mean Secchi depth data, and our second sampling event. Our second sampling event was conducted a month after our first. This was to solidify what we found. The second set of data did follow the first sets, if not exactly. Patterns observed for one snapshot in time showed to be consistent. I would like to acknowledge and thank student summer scholars, Grand Valley State University, Annis Water Resources Institute, Simon Lab, Matt Sillerhart, Emily Newman, and Kyla Sorensen.